Welcome to the Depth Dive. Today we're really getting into it. How do drugs actually do what they do inside our bodies? We've looked through your materials, and our mission here is to unpack pharmacodynamics, basically, how drugs act so you can really understand it. Think of it as decoding the conversation between a drug and your body.
Exactly. And your sources point out there isn't just one way. You've got stimulation, like adrenaline kicking your heart rate up. Then there's depression, maybe like alcohol slowing down your central nervous system. You also see irritation, I think topical creams or cytotoxic effects where antibiotics, say, target bacteria. And, of course, replacement like insulin for someone who needs it.
Okay, so quite a range, but how does that actually happen at a molecular level? Your materials mention non-receptor and receptor-mediated paths. Let's maybe break those down a bit.
Sure. Non-receptor ways are, well, sometimes more direct. Think physical action, like mannitol using osmosis, just drawing fluid out. Or activated charcoal, that just sort of absorbs toxins like a sponge.
Ah, okay, so purely physical.
Or chemical, like antacids just neutralizing acid. Simple chemistry. There you have drugs messing with enzymes. ACE inhibitors are a classic example. Or they block ion channels, like local anesthetics stopping pain signals.
Right, so no specific docking station needed for those. But receptor-mediated? That sounds different, more targeted.
Definitely more targeted. Receptors are specific proteins, usually on or inside cells. Think of them as docking stations or locks. Drugs designed to fit these can bind there. Now, if the drug activates the receptor, we call it an agonist. Morphine's a good example. It binds and produces an effect.
The key that turns the lock.
Precisely. But if a drug binds but doesn't activate, and maybe blocks the natural key, that's an antagonist. It just sits there, blocking the spot.
Gotcha. And your sources mentioned these receptors, these locks, aren't all built the same. They work at different speeds. Why does that matter?
Oh, hugely. The speed is critical. You have ligand-gated ion channels, super fast, milliseconds, think nerve signals. Then GPCRs, G-protein coupled receptors, they take seconds. Very common type. Then enzymatic receptors, like for insulin, they work over minutes to hours. Much slower, more sustained changes. And finally, nuclear receptors. These affect gene expression, so you're looking at hours for their effects to really show up.
Wow, quite a range. Those GPCRs sound pretty central, working in seconds.
But it's not just bind and go, is it? There's more involved.
No, it's often a cascade. The drug binds, activates the G protein inside the cell. That G protein then kicks off something else, often an enzyme like adenyl cyclase, which then makes second messengers like CAMP or IP3. And these molecules spread the signal inside the cell, amplify it. It's like a little intracellular relay race.
A domino effect, kind of amplifying the original signal. Okay, let's go back to agonists then antagonists for a second. The activators and blockers. Your materials mention different types of blocking.
Right. So an agonist like morphine just works, gives you pain relief. A competitive antagonist like atropine fights for the same binding spot as the agonist. If you add more agonist, you can usually overcome the block. It's reversible.
Okay, a competition. But a non-competitive antagonist is different. It might bind irreversibly or bind somewhere else on the receptor that changes its shape. So even if you flood the system with agonist, it can't produce its full effect. Phenoxybenzamine is an example there. The block isn't easily overcome.
That really highlights why dosage is so crucial. Which brings us to dose-response relationships. How do we visualize how much effect we get from how much drug?
Yeah, the standard tool is the log dose response curve. It usually looks like an S shape on a graph. The dose on a log scale is on the bottom axis, the x-axis. The response, like percentage of maximum effect, is on the side, the y-axis.
Okay, and what does the shape tell us?
Two main things, potency and efficacy. Potency is about how much drug you need. A curve shifted to the left means less drug is needed for a given effect. It's more potent. Efficacy is about how much effect you can get. Max, that's the height of the curve. Some drugs just can't produce as strong an effect as others, no matter the dose.
Potency is dose, efficacy is maximum effect.
Got it. And for safety, we look at the therapeutic index. Exactly. TI. It's a ratio. The dose that's lethal in 50% of subjects divided by the dose that's effective in 50%. LDOs over EDOs. A high TI, like with penicillin, is great. Big gap between effective dose and toxic dose. Much safer. A low TI, like digoxin, means that gap is small. You have to be really careful with dosing. Narrow therapeutic window, we say.
Makes sense. What happens when people take multiple drugs? Do they just add up?
Sometimes that's an additive effect. 1 + 1 = 2. But sometimes one drug can make another more powerful. That's potentiation or synergism. 1 + 1 = 3, kind of. And sometimes they work against each other, antagonism. That can happen physiologically, like adrenaline counteracting histamine, or right at the receptor, like naloxone reversing morphine.
And these interactions aren't just about the drug's action site, right? They can happen earlier.
Oh, absolutely. Pharmacokinetic interactions are huge. They affect absorption, distribution, metabolism, excretion, ADME, like taking tetracycline with antacids. The antacid can stop the antibiotic from being absorbed properly. Less drug gets in. Or metabolism. Some drugs speed up liver enzymes, breaking down other drugs faster, like rifampicin affecting contraceptives. Or excretion probenecid can slow down penicillin removal by the kidneys, making it last longer. Then you have pharmacodynamic interactions, where they interfere more directly at the site of action, like the synergism between trimethoprim and sulfamethoxazole.
Okay, so many interaction points. And finally, it's not just the drugs, it's the person, right? What individual things change the response?
Totally. It's never one size fits all. Age is a big one. Newborns might lack certain liver enzymes. Older adults might have reduced kidney function. Both affect how drugs are handled. Genetics plays a role. Think G6PD deficiency, or how fast someone metabolizes drugs based on their acetylator type. Tolerance is another your body adapts, especially with things like opioids. Or tachyphylaxis, which is like rapid tolerance. You see it with ephedrine sometimes. And even drug factors, like how it's given IV versus oral makes a difference. Or if a drug tends to build up, accumulate, leading to toxicity, like with digoxin.
Wow. Okay, so it really is a complex dance. How the drug acts, how the body handles it, the dose, other drugs, the specific person, all feeding into the final effect.
It is. The precision involved at the molecular level is pretty stunning. And the ripple effects on our overall health are just immense.
It makes you wonder, doesn't it? Given all these known factors influencing drug response, what other, maybe more subtle individual biological differences might be playing significant roles we haven't fully grasped yet? What new avenues does thinking about this complexity open up for you regarding truly personalized medicine down the road? Something to ponder. Thanks for joining our deep dive today.